Hello again and welcome back. So in this episode, we're going to look at detecting cyanobacteria and detecting cyanotoxins. So I guess to kick things off, if I want to see if I have cyanobacteria present, is there anything I can look for in water? So any visible signs?
Yeah, so I guess the first thing you sort of are looking for is some discoloration in the water, maybe a little bit of a greeny tone, maybe something's changing with your turbidity, maybe you actually have sort of a fluorescent probe. So there's these sensors now which sort of detect the fluorescence from either chlorophyll or a compound produced specifically by cyanobacteria called phycocyanin.
Okay.
And so if these basically you can detect quite low levels of those pigments in your water supply. So chlorophyll, obviously that could be produced by both algae and cyanobacteria. And that's why sort of these phycocyanin sensors can be quite good, as if you're worried about cyanobacteria and not so much about algae, then they're really beneficial because they're specific. But, sort of low levels of cyanobacteria, you're not going to be able to necessarily see with your naked eye. And so that's when you collect a water sample and you send it off to Cawthron to analyse.
And so what we do once they arrive at the lab, what we do with those samples is that we analyze them by microscopy. So we literally look at them and we will be able with that to see and identify, but also count the cells in the sample, and that gives us an understanding of what's in there and in what abundance as well.
Oh, so it can be done quite quickly then, from taking the sample to identifying the risk.
Yes, that's right. But depending on the number of samples we have in the queue at the lab, so in a busy season, sometimes it can take, a couple of days before we get to that. But hopefully, if we were to have a sample taken to the lab, it takes a couple of hours if we have to do the full enumeration and identification. So it's relatively quick.
Oh, that's pretty quick.
Do the samples need to be alive? Like, do they? How do they?
That's a good question. So if in the sample there might be some grazers, the cells can multiply and alter the nature of the sample when it came out of the field. So what we recommend is that they are preserved upon taking.
Right.
So a sampling officer would go scoop the water up and add some Lugol's iodine, which is the preservative we recommend, and then send it to us at the lab. And then that ensures that the numbers of cells aren't altered by the time it takes to transport and natural processes that happen in the sample.
Makes sense.
But even if the sample could not be preserved in the field and it arrives to us overnight, that's okay. We still can work with that.
Yeah. So if I see a nice green scum in my my river and I scoop some up and all good to send it to you guys.
We could deal with that.
Yeah. So I guess other sort of visual signs as well, I'm guessing like algal growth on rocks, things like that. So I'm thinking like just before you get down to the sampling of the probes, any kind of visible identifiers that you might have a problem.
Yeah, so you sort of imagine you're operating with a river water supply or a stream. So what you're looking for there is looking at the base of the river and you're looking for these mats or sort of slime, people might call it, growing on it. And so Microcoleus, which is our most common benthic cyanobacteria, so a type of algae which grows on the base of a water system, so either the bottom of a lake or a pond or on the base of a river, usually on sort of stones in the bottom of the river. The most common one that we have here is called Microcoleus, and that sort of looks sort of brown, black coloured and looks quite different from sort of green algae, Spirogyra, the long filamentous algae that you might have in a river, or sort of a diatom mat, which will be sort of quite a light brown or sort of a little bit fluffy even. But it is really hard when you're just starting out on your sort of cyanobacteria monitoring journey to be able to sort of go down, look at a rock, say, oh yeah, that's Microcoleus, that's Cyanobacteria, there's some green algae, here's some diatoms, particularly the diatoms in the Cyanobacteria. So when you're in that situation, what you want to be doing is to collect that rock, pick it up, and then scrape it off into a tube. And then if you send those scrapings to Cawthron and the team, what they'll do is they'll go and they'll put those under a microscope, and they'll be able to look at the morphology and the size and go, yep, that's Cyanobacteria. Nope, that is just some good old slime from the bottom of the river.
All right. So then once you've identified cyanobacteria, you can go in and look at the genes and see if that's a greater risk.
Yeah, exactly. Yeah.
So I guess a lot in what we've been talking about today, we talked a lot about rivers. So I guess that brings me on to my question is, are rivers the only source at risk? What other water sources would you deem at risk from cyanobacteria?
So we kind of break cyanobacteria down into the sort of benthic cyanobacteria, which is growing on the base of a water supply, and planktonic cyanobacteria. So this is cyanobacteria that are floating around in the water column, the one that's causing John lots of troubles to try and get rid of because they're far too buoyant. So in sort of lakes and reservoirs would predominantly see planktonic cyanobacteria, unless you've got a river which is joined to a lake where some planktonic cyanobacteria are living and they're flowing down through the river. And then in rivers, we'd usually see benthic cyanobacteria growing on the base, and that's just because a river's flowing. If you want to have something live in there for a long period of time, it needs to be attached to the bottom. But you can see benthic cyanobacteria in lakes and reservoirs as well. And so these are sort of, you know, they're sort of a really annoying threat for water suppliers. Because quite often you can't see them. So, you know, sort of a planktonic cyanobacteria growing in the water column, you pick up your sample and you can see the cyanobacteria when you look at it under the microscope. But the benthic cyanobacteria attached to the bottom, you know, unless one of those mats detaches and floats up to the surface, ends up over on the shore, you won't necessarily know that it's there. And so that's when looking for things like those musty aromas, the earthy undertones in your water supply, and you don't have other cyanobacteria to explain it. That's when you maybe need to think, okay, do I have benthic cyanobacteria present? And then it gets a little bit tricky. We're sort of looking at sort of dropping cameras down into the bottom of the reservoir. We're sort of sending divers in to try and see it. Or we're sort of taking these sediment grab samples where we've got a claw and it goes in and it pulls up some of the mud from the bottom. And we have a look around in there to see if we can find cyanobacteria. But a good practice is if you do have a reservoir or you're drawing water from a lake for drinking water, to be scouting around the sides of the lakes and actually looking for mats which are floating up on the side. So, you know, if you go and you see one, what you'd want to do at that point is to sort of collect a sample of that, to chuck it in a tube, and then if you send it into Cawthron's lab, they look it under a microscope and figure out if that's cyanobacteria present in that mat, or, you know, whether it's just other slime. So similar scenario to the rivers there.
I mean, a big part of monitoring a treatment plant on a river is making sure that you check your intake and, you know, that there's no giant mats of detached bacteria and the intake being sucked into your treatment plant.
So if you've got a screen and you're starting to see those mats there, you know, that's sort of the time when you want to be taking those samples, seeing what's actually growing in that mat, because it might just be algae, or it might be cyanobacteria, or it might be a non-toxic cyanobacteria, and you just don't know by visually
looking at it.
Picking it up.
So I mean, so lakes and rivers, what about bore sources? Is there any time that a bore would be at risk?
So bore might be at risk if you have sort of a water supply nearby which has lots of cyanobacteria. So you sort of imagine that you've got a river, it's got lots of benthic cyanobacteria in it that are potentially producing toxins, and your bore, your groundwater source, is actually quite close.
Yes.
And, you know, there might be some connection between the two of them. So in those scenarios, you're sort of just trying to take a precautionary approach to figure out whether this risk in a different water supply is actually infiltrating your water supply.
Yes. Yes.
And what we might do in that scenario, because the cyanobacteria themselves are unlikely to be getting through all of the sand and soil to get into your groundwater supply, we'd be looking for the toxins there.
Yes.
And we'd probably use a technique that we call spat sampling. And so basically this is sort of a resin which we put in a little net and we drop it down into the water supply, and that resin starts to bind up the toxins.
Okay.
And so it's sort of this great concentration effect that we get. So we might leave it down in the bore for a week, pull it back up, and then we take that back to the lab, extract off the toxins or maybe no toxins. That's the ideal scenario from that resin, and then we analyze that. And so, you know, sort of if we just went in and tested the water straight, you know, we might find out, you know, there's below worrying levels of cyanotoxins present. But using these spat samplers, what we get is a bit more information about whether there is sort of a potential connection between those two waterways. And it gives you sort of a greater sense of assurance that actually this waterway, this bore source is all good. You know, it's not affected by these cyanotoxins over here nearby. And, you know, we can feel comfortable and sleep easy at night.
Because that makes a lot of sense. I mean, you're using the spat bag, you're testing something that's directly coming from the river into your bore and shouldn't really be coming from anywhere else, right? Because, you know, speaking from an engineering perspective, trying to establish whether there is or isn't a connection between a bore and some other water source nearby, sometimes it's obvious. You know, in the kind of conditions where, say, the turbidity rises and falls with, you know, the heavy rain causing the river level to rise and fall and maybe speeding up the passage of the water through, or something like conductivity changing whenever it rains, you know. But it's very difficult to know for sure, you know, unless you do some very, very sophisticated tracer testing type approaches. You might get as far as thinking that you're pretty confident that, you know, something is happening, but you'd probably never be 100% right. Whereas if you measure the toxin, you are literally testing for the thing that's, you know, of concern.
Yeah,
in that case.
and by increasing the sensitivity of our testing, or sort of how low we can see, it just gives us that extra confidence, you know, that there's something that we haven't been able to predict.
Indeed. We all know that the atomic age testing is not always representative, and, you know, of a bore's transit times in certain conditions, and really that's the one that can be problematic, I guess. Is the atomic age only represents what the bore was like when it was tested, and depending on what might happen when it isn't being tested, you can get completely different effects on the bore. So effectively short-circuiting occurring.
It forms part of a risk understanding approach, you know, with the update to the drinking water standards, July 2022. Understanding the risk profile of your source water is going to be very important. And if your source is at risk of cyanotoxins, you're going to have a cyanotoxin response plan with sampling happening periodically, a monitoring plan and response and actions, so that you protect the consumer from cyanotoxins.
And the crux of it is whether the supply is directly influenced by a river, isn't that right?
So how connected it is.
So how do you prove that, or not, as the case may be?
So without getting into cyanotoxin testing, which, you know, is relevant if you've actually got a cyanobacteria bloom in a water supply nearby, is there any other ways that we can sort of figure out connection between a groundwater source and nearby?
I think that probably links back to what you were saying earlier about, so when you get heavy rainfall or something like that, if you see the quality of water in your bore changing, then it's kind of an indication that it's quite closely connected. So I guess if you see external events such as rainfall causing effects in your bore, then you have a pretty good indication that there's a connection there somewhere.
That's right, and under the new drinking water standards, there's a duty of care that's been basically instilled into the document. But one of the key things is understanding your source, and explicitly that you go and you check your source during extreme weather events. So that means drought or heavy rain. So at least from a pure chemistry perspective, let's say, of pH and conductivity and maybe alkalinity or something like that, you go out and you check whether something strange is happening. Because if something strange is happening, you at least know that it's happening, and that may imply that yes, it's connected somehow to another water source, even if you can't go out there and prove it 100%. So it's about managing the risk, because if there is potentially cyanobacteria in a connected river and you think that it may be connected, then you should plan for that.
So I guess, that kind of brings me nicely on to my next question around risk. Is there a time of the year when the risk is greatest? So, in peak of summer and the depths of winter, am I at a higher risk of cyanobacteria and cyanotoxins, or is it fairly consistent throughout the year?
Yeah, so summer times when we've got these warmer months, cyanobacteria in rivers definitely start to take off. So cyanobacteria in rivers, these benthic cyanobacteria, because they grow on the base of the river, when we get sort of a big rain event and lots of water coming through the river, they get washed out. And so we have this resetting of that environment where they're all gone, and then they'll start to grow again from that point. And just in summer, we have longer periods between each of those resets. So basically, if you'd have one of those resetting events, the longer amount of time that's gone in between,
the greater the
likely
population
that there's going to be cyanobacteria present. And sort of in lakes as well, where water's a bit warmer in the summer, and there's more daylight coming through, it's just more likely that cyanobacteria is going to be present. And what we often see is that the sort of community of cyanobacteria will change throughout that sort of big season. And so, you might actually have cyanobacteria throughout that whole sort of summer period, but they're not necessarily going to be the same cyanobacteria. So this is where it's really important to keep monitoring over those months to know what's happening with that community.
And understand what's changing.
Because at the beginning you might have sort of a non-toxic species, but eventually that's going to be replaced by a different type of cyanobacteria in your later season, which might be a toxin producer.
And those are times of great risk, right? I mean, when it rains heavily after a dry period, and those cyanobacteria get ripped off of their happy home in mass quantities and go straight to the water treatment plant. Or if you've got a lake and it gets warm enough to what do they call it, stratification, and then the overturn that can happen and massive change in conditions and upset the balance basically.
Yeah, so that's the thing I sort of forgot to mention as well. In these lakes and reservoirs, if we're getting stratification occurring, some really cool water chemistry or nasty water chemistry dividing, depending on which side of the fence you're sitting on, can start to happen. And so in those warmer months, you can have the lake separating out into two environments. So at the top and the bottom, they're actually separated, and that's what we call stratification. And what happens is down in the bottom part of it, that gets anoxic, so there's not enough oxygen. And there's types of bacteria, microbes, that can live in that anoxic environment, and they'll start to release nutrients from the sediment at the bottom of that lake or reservoir. So this actually provides sort of a fuel source for cyanobacteria and algae to grow on. So when we actually get that turning over, so you've got a long, stable, hot period, the bottom community down here, they've been releasing nutrients, but there's no mixing in between the two of those. You've actually got this really nutrient-packed environment at the bottom of the lake, and then you get a rain event and the temperature changes and we get mixing, those nutrients then become available to the top of the water column.
It was like dumping a ton of fertilizer into your dam, something like that.
Yeah, exactly. Yeah, you sort of just gone and given them everything that they need to take off if they get a good growing period. So that's kind of why the old cyanobacteria guidance that was out there for drinking water supplies, they had sort of quite a few stratification of a lake was one of their sort of key things to be looking out for.
Yes.
And that's the reason why.
Yeah.
Oh, that makes a lot of sense. I mean, like say, at the bottom, they're literally producing food for the top, and then once you get that mixing, you can get those rapid blooms afterwards. So yeah. Okay, I think we've answered probably quite a few of the questions I had here. So one of them was around sort of sampling and testing. So we've spoken a lot about sort of spat testing. We've spoken about taking sort of grab samples and sending it off to your lab. Is there any other types of testing that perhaps Cawthron do that we should know about?
The three main ones really are the microscopy testing, so that's the visual assessment, then the gene test to see whether there are potentially toxic ones in the sample, and then the third one, and importantly, is the actual toxin testing.
Yes.
So that's done using some machines, so they are high-tech machines that allow us to detect whether there are toxins in the sample, and it allows us to measure them very accurately as well. So the machines are calibrated with standards.
Yes.
And they are purified versions of the toxins we're looking for, and it allows us, so knowing those measures and comparing a wild sample to that, allow us to know exactly how concentrated that wild sample is.
Yeah.
And so that's the determinant whether there are toxins in there or not. That's the one that is the real diagnostic. And that's used for regulations.
Okay.
Excellent.
Interesting.
Another good thing to note here is quite often the lab, they're going to say, it'll be, if you're sending us a sample, send us a preserved sample and a non-preserved sample. And the reason that we have that in there is because we have this tiered approach to testing. Where we'll go in and we'll look by microscopy, see if there's cyanobacteria present, see if there's potential toxin producers present. That might move on to the gene testing. Do we actually have cyanotoxin production genes present? And if that's a yes result, then we'll move on to the toxin testing afterwards. So that means you don't jump in with, the most intensive testing right at the beginning. We sort of move through, we understand the potential risk, the probable risk, and if there's an actual risk. And so to do that gene screen and the toxin testing, what we need for that is a non-preserved sample. So if we get both of them right from the beginning, it means there's no sort of waiting period in between each of those testing steps. We can just go bing, bing, bang, boom, and you get results a lot quicker.
And that sequence of testing can be customized as well, because it's not all customers that will want the same type of testing, depending on the situation. So we're really happy to customize the way that we will go with that combination according to their needs.
Yes, I mean, it makes sense. It's at least from a water supply perspective, you probably don't start directly testing for the toxins, because that would mean that you're worried that there's actually toxins in the supply. So you shouldn't really be at the point of
and
checking for the bad stuff.
If you don't have cyanobacteria, then you're not actually going to have toxins. So that's why there's sort of a staged approach, both in sort of the guidelines, but also in sort of what we offer to clients as well.
Yeah. Now, Brett, I know you've done some quite interesting work with some online instrumentation. I know Jonathan's quite passionate about this as well. So, I mean, outside of the testing, is there any other ways that we can, use spectrometry or something similar to even just get an indication on when things are changing? So once you've understood your water source, there's a risk. Can we use anything else as like an early warning indicator?
Yeah, you can. I think there are conventional methods and the more advanced spectrometry. So spectrometers can look at the response, usually like JP was saying, with the luminescence. But but that requires calibration. It does depend on how things change, and so it would be source dependent. But there are also some more conventional methods. So within a particular lake of a water supplier, it's quite deep, and depending on where you take the water from in the water column, the algae or the cyanobacteria can affect even the pH. So within the water column within the middle, the pH in this instance I have in mind was much higher than below and above. And so, and also you get the floating algae and the benthic ones on the bottom of the lake. But the spectrometers that's a very interesting area. There's a lot of work being done there, and it is source dependent and requires calibration. And as JP was saying, just seasonal changes will affect the responses that you get.
Yeah. I mean, it's talking about the kind of tests that you guys do in your lab. There's always going to be a lag time, isn't there, between the sample getting taken and the actual placing it under the microscope and having a look. So,
and getting your numbers back.
Exactly. So it's one of those things where having a little bit of foreknowledge can be quite powerful. But as Brett said, there's not really any easy parameters that you can look at. And you mentioned the fluence and the illumination methods. Directly seeing that there is maybe something there, but it may not be of a worry. At least in terms of going back to understanding what your source is, it might be more than one parameter at the same time. Things like the pH swinging with the 9-day cycle significantly. A decrease or an increase in the amount of alkalinity because of the species, doing various things, or the nutrients, for instance, in the lake being released. Say ammonia might be a good one when the lake overturns. Any one of them really just indicates that something is changing, and not in the kind of way where you think, this will make the water harder to treat. You're not talking turbidity or organics directly. You're talking about something that there's a difficult to quantify risk. So it's at that point that you might want to start and go getting these tests done because something is happening, but you don't know exactly what.
Yeah, there's some changes happening to the water chemistry, but we're not entirely sure what.
And it might be part of your monitoring plan where through historic sampling, you might say these are conditions where we know there's a higher risk, or we think there's a higher risk, and then you would up your monitoring and your sampling for certain tests that you won't be performing throughout the year, but you might perform them when it's warmer or when there's there's going to be a turnover.
Yeah,
and some work that Brett has done is to look at different parameters and create a sort of surrogate. So something that doesn't necessarily mean the concentration of cyanobacteria or even that there is cyanobacteria. It's just that your sources change significantly. There's now an increased risk, and using the kind of the AI neural net methods and various other things, you can basically assign weights to the parameters, and when those weights all come together, not one particular thing, but all of them at the same time, that something is different. That may correspond to tests that you've previously done that say, yes, we've got cyanobacteria again.
And I guess the more you actually sort of invest in building up that knowledge on your water supply, the better you are down the track. If you can actually explain what's leading to changes, the next time you have that occur, you can either know to be responding, or you can feel a bit more assured that it's because of this thing, and you go and check it out, and that's all good.
Excellent. Well, I think that's probably enough on detecting cyanobacteria and cyanotoxins. So thank you for that, and we'll come back for the next episode shortly.